Let's move our discussion on to cardiac muscle. Cardiac muscle is very similar to skeletal muscle, but certainly there are some differences that we will cover. Before we do that, let's just talk about the basic circulation of blood from the heart and to the heart. To be clear, the heart's main purpose is to pump blood throughout the body, which is known as the systemic circulation. So the left ventricle, which is roughly right here, and to be clear, I should say we're speaking in standard anatomical position, so that is the specimen facing you. This is my left hand, but I realize to you guys looking this direction, that's probably your right side. Anytime we're talking about anatomy, we are referencing as in facing you. So to be clear, the majority of the heart, if we're holding a human heart, we're looking at ventricles. The atria, especially the left atrium, tends to be on the back side of the heart. And we'll take a closer look at the orientation of these chambers, the two atria and the two ventricles in a moment. But I just want to once again get back to talking about circulation. There is the systemic circulation, which is oxygen-rich blood being distributed throughout the whole body. It's being pumped by the left ventricle. That is to say, the left ventricle is creating a pressure gradient to allow blood to be ejected from the left ventricle into the largest artery in the body, which is the aorta. The aorta is then going to bifurcate or split and split again and split again into smaller and smaller arteries, into arterioles, making its way to capillary beds at all the tissues in the body. So the systemic circulation, once again, is providing oxygen-rich blood to the entire body. So we're talking the big toe or big thumb, depending on what that is or how you perceive that, the brain, the GI tract, the pancreas, the muscles, the kidneys, all tissues in the body, because all tissues in the body are going to require oxygen and nutrients to keep them running. And to be clear, the heart muscle, the myocardium, is also one of the tissues that the heart itself is delivering blood to. These are coronary arteries, and I realize this is a bit hard to conceptualize, but the actual heart is pumping blood to the heart muscle, and we'll get to that in a subsequent video. So systemic circulation is pumping blood system-wide, whereas the pulmonary circulation is pumping blood merely to the lungs. The right ventricle is a pump for the pulmonary circulation. To be clear, the lungs are not here. The lungs would actually be on each side of the heart. The heart resides in a space between the right and left lung, known as the mediastinum, which is within the thoracic cavity. But once again, the right ventricle is pumping oxygen-poor blood to the lungs to drop off carbon dioxide and to pick up some more oxygen. That oxygen then proceeds back to the heart into the left atrium. Left atrium then drops that blood into the left ventricle, pumped throughout the whole body, providing oxygen and nutrient-rich blood to all the tissues. And then the oxygen-poor blood, after it is dropped off that oxygen, picked up waste products, returns back to the heart via the inferior and superior vena cava, which I do not have drawn in this image, into the right atrium, and then from the right atrium, it drops into the right ventricle, and we go through this cycle again. To be clear, the right side of the heart, right atrium and right ventricle, is often described as oxygen-poor blood, because indeed that's what it has, and the left side of the heart is oxygen-rich blood. So let's take a closer look at these chambers and the pressure gradients that allow all this to happen. Here's another depiction of the heart: right atrium, right ventricle, left atrium, left ventricle. I have them color-coded differently just so you guys can differentiate between them. To be clear, the left atrium, not this much of the left atrium is not visible on the anterior version of the heart. And if the heart is not cut open, this would actually be a coronal section of the heart. But the left atrium is more posterior, if not entirely posterior, than anything else. But we tend to draw it, or at least I tend to draw it this way, just so we can show the left atrium, because I can't show anterior and posterior at the same time. Right atrium, right ventricle. Right ventricle is going to pump oxygen-poor blood via the pulmonary arteries to the lungs. I'm not showing the pulmonary artery here. Comes back to the heart via the left atrium, or to the left atrium, to the left ventricle, which pumps oxygen-rich blood out the aorta throughout the whole body. That's the systemic circulation. And then it returns that oxygen-poor blood to the right atrium into the right ventricle. and that keeps going. So for all of this to happen, we need to consider pressure and pressure gradients. Air and water always flow down pressure gradients to an area of lower pressure. We could say from high pressure to low pressure, or high pressure to lower pressure, but really all you need to suggest is that blood, in this case, is flowing to an area of lower pressure. And I only make that qualification because it may not necessarily be leaving an area of high pressure. Obviously, these pressure numbers are relative to different regions in the body, but there can be regions in the body downstream that actually have fairly low pressure, but it's still moving blood to areas of lower pressure. So high pressure would be roughly 120 millimeters of mercury leaving the heart. When we get back to the heart, it's probably in single digits. So as we're looking at this number right here, 120/80, you may recognize that as being the fairly standard blood pressure. 120 is when the left ventricle is contracted, 80 is when the left ventricle is relaxed, and we'll talk about all of this. Contracted is also known as systole or systolic pressure. Relaxed is diastole or diastolic pressure, and pressure is measured in millimeters of mercury. Let's talk about some basic physics with regards to pressure and volume. And when we're talking about volume, we need to distinguish between the volume of the chamber versus the volume of the fluid. So when we're talking about a chamber or container, as I've written here, for us, that's applying to the chamber of the heart, any of the atria or any of the ventricles. It certainly can be referring to a blood vessel, because blood vessels are containers too, that vasoconstrict and vasodilate and modify the size of that container. As we look at this, we can see an increase in volume of the container is going to result in a decrease in pressure. So if we have the left ventricle that gets larger, the pressure within the left ventricle is going to decrease. If the left ventricle gets smaller, that happens due to ventricular contraction or ventricular systole. As the volume gets smaller, pressure increases. So when we're talking about the volume of a container, the relationship between volume and pressure is inverse. That is to say, as volume goes up, pressure goes down. As volume goes up, pressure goes down. And as volume goes down, pressure goes up. If we look at the volume of the fluid, it is a direct relationship. That is to say, an increased fluid volume is going to correspond to increase in pressure. A decrease in fluid volume is going to correspond to a decrease in pressure. That is a direct relationship. So if you can think of a water balloon, if you keep adding water to that balloon, add fluid, add fluid, add fluid, at some point, as long as there's a seal at the top on the water faucet, that balloon is going to explode or pop. And that's due to an increased fluid volume, increasing pressure. The balloon pops once the pressure in the balloon is greater than outside, and then the fluid's going to move from an area of higher pressure to an area of lower pressure. Now, the same thing, we can apply that same analogy with the water balloon to the volume of the container. That is to say, you have a water balloon, it's filled with water, the knot is tied on the top, we're not adding more water. But if we squeeze the water balloon to decrease the size of that container, then eventually the pressure is going to get so high that the water balloon explodes, because water's trying to get to an area to lower pressure outside the water balloon. So we need to be mindful of the relationship between volume and pressure when we're trying to understand the flow of blood plasma throughout the body. And once again, we need to be mindful of whether the change is happening to the container, i.e. one of the atria, one of the ventricles, or the blood vessels themselves, or there's a change in fluid. For example, if anyone ever has a severe bleed, they are hypovolemic, they have low blood plasma levels, that's going to decrease their pressure. And the reason that's a problem is because we need pressure gradients to allow blood to flow throughout the body to provide nutrients and oxygen-rich blood throughout the body. Okay, with that said, let's look at some examples of the left ventricle and a little bit to the left atrium. So to be clear, what I am demonstrating in all these images here, on the bottom is the left ventricle. It's the same ventricle. It just at different stages. And on top is the left atrium. Here I'm showing end systolic volume. That is to say, the volume of blood in the left ventricle after ventricular contraction. And once again, we're really talking about the left side. The principle applies regardless of what chamber we're in. But at the end of ventricular contraction or ventricular systole, which is the same thing, there is some residual blood left in the ventricle. So end systolic volume is the volume of blood left in the left ventricle after one contraction. Now, during this time, blood is flowing from the left atrium into the left ventricle down a pressure gradient from an area of higher pressure to an area of lower pressure. Certainly, most of this is happening due to gravity, but blood in this situation, blood is flowing from the left atrium into the left ventricle. Now, as fluid or blood plasma starts to increase in the left ventricle, that's an increase in fluid volume, which is going to correspond to an increase in pressure within the left ventricle. And once that pressure is high enough, or at least equal to the pressure in the left atrium, blood is not going to keep dropping into the left ventricle. What needs to happen in the left atrium to ensure the remaining blood gets into the left ventricle is atrial systole, atrial contraction of the atria. By decreasing the volume of the container, we're going to increase the pressure in the left atrium, and that moves into here. This is during ventricular diastole, that is to say, the heart is in a relaxed state. It is not pumping. At the very end of diastole, just before contraction or just before systole, blood is remaining in the left ventricle. In theory, there's no more blood in the left atrium moving down here. And the reason that blood is not being ejected out the aorta right here is because at this moment, pressure is still higher in the aorta or in the systemic circulation than it is in the left ventricle. I want to point out, this is the aorta. The aorta does not depart the left ventricle here or right here that I'm showing. It departs the left ventricle as such. I'm only drawing it here to simplify this drawing right here. So this is end diastolic volume, the total volume or amount of blood plasma in the left ventricle just before it contracts. Ventricular systole is going to happen when the left ventricle contracts or gets smaller. The volume of the container gets smaller, pressure is increasing, and then once the pressure is greater in the left ventricle than in the systemic circulation in the large arteries like the aorta and the major arteries coming off the aorta, then blood is going to be ejected from the left ventricle into the aorta. So what I hope you've noticed is that pressure is increasing in the left ventricle two ways. One, by an increase in fluid volume. Increase in fluid volume corresponds to an increase in pressure, and then a decrease in size of the container. The volume of the left ventricle is also going to increase pressure. So pressure is increased two ways: increased fluid volume and decreased size of the container. So how much blood is ejected from the hearts? Well, that is what's known as cardiac output, the amount of blood ejected from the left ventricle per unit time, which generally is liters per minute. And we can determine cardiac output if we know heart rate and stroke volume. Stroke volume is the volume of blood ejected from the left ventricle after one contraction. So if we know heart rate and stroke volume, we can determine cardiac output. Now, stroke volume is EDV end diastolic volume, that is the volume of blood in the left ventricle immediately before contraction, and then subtract end systolic volume, that's the amount of blood remaining in the left ventricle immediately after contraction. Before contraction, after contraction, the difference represents how much has actually been ejected from the left ventricle after one contraction, otherwise known as stroke volume. Heart rate times stroke volume gives us cardiac output. So I'm just making up some numbers right here. If end diastolic volume is 100 milliliters, end systolic volume is 20 milliliters, then stroke volume is going to be 80 milliliters. That is the difference between 100 and 20. And let's assume heart rate is about 70 beats per minute. So let's do some math here to try to figure out, to see if we can determine what cardiac output is. Okay, so we can cancel out some things, and those things are beats, and this is going to leave us with 5,600 milliliters per minute. But because we already said cardiac output is generally reflected in liters per minute, we need to convert milliliters per minute to liters per minute. And now we can cancel out milliliters and cancel out some of the zeros, and we end up with 5.6 liters per minute is cardiac output, and that's fairly accurate. Our entire blood measures roughly 5 to 6 liters, so this is suggesting that the left ventricle is pumping over the course of 1 minute all our blood throughout the systemic circulation, once again over the course of 1 minute. That is cardiac output. Now, I have already said that the myocardium, or the heart muscle, is very similar to skeletal muscle. So let's take a look at this at the cellular level. What I am showing here is a myocardial contractile cell that I often abbreviate as an MCC. The myocardium, myo means muscle, cardium refers to heart. The myocardium is composed of two types of cells: the contractile cells, myocardial contractile cells, and myocardial autorhythmic cells. The myocardial autorhythmic cells set the rate or rhythm of the heart, that is to say how often it beats and contracts, and we'll deal with that in a subsequent video. For here, we're going to talk about the MCCs, myocardial contractile cells. Once again, I tend to abbreviate these as MCCs, and these are stimulated to contract from the MACs. So anatomy for these MCCs, very, very similar to skeletal muscle. We have a sarcoplasmic reticulum right here. We have a protein channel right here, which is similar to the DHP receptor, but different. DHP receptor is a voltage sensitive protein. This right here is a voltage sensitive calcium channel. We have a ryanodine channel that allows calcium to leave the sarcoplasmic reticulum. The big difference here is in skeletal muscle, the ryanodine channel is a mechanically gated channel where we have that protein or chain pulling on the ryanodine channel to open up, whereas here, this is a ligand gated channel. The ligand that opens this is calcium, and that I realize is going to be confusing because we're talking about the release of calcium from the sarcoplasmic reticulum. This is known as calcium-induced calcium release. The calcium that is coming in here is coming from the extracellular fluid. This calcium is green, that calcium is brown. Calcium is calcium, they don't have different colors. I have just made them in different colors so we can follow the pathway here. So I'll go into the details of this in a moment, but to be clear, if we look over here, this is a myocardial contractile cell in the relaxed or non-active, non-excited state. It's polarized, negative charge. There's no impulse being directed from the MAC to the MCC. The ryanodine channel is closed, no calcium is being released, and this voltage-gated calcium channel is closed. So when the MACs depolarize, that depolarization event is going to be distributed to the MCCs, the contractile cells. That depolarization event is going to proceed down the T-tubule and open up this voltage-gated calcium channel. That's going to allow extracellular fluid calcium to move through that channel and to bind to the ryanodine receptor channel on the sarcoplasmic reticulum, open up that channel, allow calcium to depart the sarcoplasmic reticulum and go bind to troponin, which then moves tropomyosin out of the way, and boom, we get muscle contraction. Certainly I left out a lot of steps of the sliding filament theory there, but look at the previous video on that if you need a review. Like skeletal muscle, there are pumps returning ions back to where they came from. So there's going to be a calcium ATPase that pumps calcium back into the sarcoplasmic reticulum to maintain calcium's high concentration in the sarcoplasmic reticulum. There's also going to be a sodium-potassium pump that restores the chemical or maintains the chemical gradients of sodium and potassium. But we also have to return this extracellular fluid calcium back to where it came from, and that's in the ECF. That is achieved via an antiporter using the kinetic energy of sodium, an antiporter known as the MCX, sodium calcium exchanger. Calcium is going to be pumped out of the cell against its gradient in exchange for sodium, which is moving in passively. The passive movement of sodium into the cell creates the kinetic energy needed to move calcium out of the cell. This is secondary active transport. It is an antiporter because calcium and sodium are going opposite directions. The ratio here is 3 sodium come in down its chemical gradients and 1 calcium leaves. Now, the 3 sodium coming in potentially could cause problems. That is to say, it could cause the cell to depolarize when we do not want it to depolarize. We only want the contractile cell to depolarize when the myocardial autorhythmic cell dictates so. That is to say, when an impulse is transferred from an MAC to the MCC, that is when we want depolarization, not to this sodium coming in, which is used merely to pump calcium out of the cell. But fortunately, these 3 sodium that come in immediately get pumped back out of the cell via the sodium potassium pump. We'll talk more about the myocardial autorhythmic cells and how tetanus is avoided within the myocardium.